Yeah, so like Linda said, I'm really happy to be here today. I'm a HIV doc at UCSF, and I've been working in our HIV cure research program for almost 7 years now. Time really has flown by. And so I made a bunch of slides about, I think, my perspective on considerations related to long-term survivors with HIV with a focus on age and comorbidities. I think a few people aren't muted, so if everybody's just able to mute, that would be great.
Yes.
Awesome. Okay, so as was mentioned, I'm part of the SCOPE program at UCSF, which has been in operation for over 20 years now and follows thousands of people living with HIV. And over the last 10 years, we've had a growing focus on HIV cure-related clinical trials. And so I've worked on a number of cure-related trials, including a quite complex one that I will show over the last few years, and we've learned a lot from that. But first, some background. As many of you know, the main barrier to curing HIV to date has been the existence of the HIV reservoir. And so we know that HIV meds, ART, work really well. But when people stop their HIV meds, their HIV reemerges in a very predictable way, and that's shown in this graph on the left. So these are people who stopped their meds, and then over 2 to 4 weeks, the virus reemerges in blood and spreads quite quickly. And the reason for that is that the vast majority of HIV in a person's body is sort of hiding out in a state of latency or dormancy in places that are pretty difficult to find. So when we're measuring HIV in a person's blood, we're really just measuring kind of the circulating HIV that's floating around. And that's what becomes undetectable on testing. But the vast majority of the virus in a person's body is actually hiding out in cells that are either in the blood or even more common in the tissues. So what we can typically measure in clinic is really just the tip of the iceberg of the virus. And so because of this, most outcomes in HIV cure-related studies involve what's called an analytic treatment interruption, or ATI. And what that means is that after an intervention is performed, the study volunteer is asked to pause their HIV meds, and they're monitored really carefully over time, clinically, make sure they're feeling okay, but also with laboratory tests, including CD4 count, viral load, and a variety of other safety tests. And we sort of watch and see whether the virus comes back, and if it comes back, how quickly it comes back, and how high it goes. So in all of these studies, what is expected is that the virus will come back and sometimes will kind of come under control. But the only way to figure that out is to go off of meds, which in real life, we tell people never to do. So most cure studies so far have focused on what's called immunotherapy, and that just means therapies that sort of alter or manipulate the immune system to make it better at fighting HIV, either clearing HIV or controlling HIV. And so I've listed on this slide all sorts of different agents that are under investigation, and there are all different categories of agents. We don't really need to go into that now, but all of these ultimately have an effect on the immune system in a person. In addition, studies so far of single therapies, each one of these in isolation has not really shown huge beneficial effects. And now there are more and more efforts to combine immunotherapies to try to cure HIV, just like we combine HIV meds to control HIV. And then on top of immunotherapy, I think what is happening now and what is coming over the next 10 years are even more sophisticated immunotherapies, like cellular therapies, like CAR T cells, which you may have heard of, or gene modifying therapies where we actually change the genetics of a cell or of a person to try to get them to better control or clear HIV. So this is an example of an immunotherapy trial that some on this call were involved with, advising and scientifically. So this was a study that we completed at UCSF between 2020 and now, funded by AMFAR, where we used a combination of immunotherapies to try to induce an HIV cure or control of HIV. So we gave a series of HIV vaccines to alter T cell responses in people with HIV the first 12 weeks of the study. Then we gave a booster vaccine, like a super immunogenic booster vaccine, after 20 weeks. Then we gave broadly neutralizing antibodies, which you may have heard of BNAbs, which are kind of a IV and intravenous infusion of anti-HIV antibodies designed in the lab. Then we gave a toll-like receptor agonist, so another immune system booster for 10 weeks. Then we gave more antibodies. You can see how complicated this was. And then after all of that, we stopped people's HIV meds to see what happened. So I'm not going to talk about the results of that here. I presented them at CROI, and I'm going to present them next Saturday in Australia. But we think we had an effect, but look at all of the things we had to do, and look at all of the things that people had to be willing to go through to potentially have that effect. So quite complicated. So I think that there are both safety concerns and efficacy concerns related to HIV cure trials and age. And I'll talk about each of those. I'll talk mostly about the safety concerns, but I will at the end mention some of the efficacy concerns as well. So there are a lot of risks with participating in HIV cure research. There are both risks to the participant, to the person who's participating in the research, and there are risks to others. So the risks to the participant are, there's obviously a risk of doing all of this experimental therapy. Each of those drugs, comes with risk. There's risk of stopping the HIV meds. There's risks of, getting measured in 1,000 different ways and poked and prodded and coming into the medical center and all sorts of other tests that are done. And there are risks related to COVID, which I'm not going to talk about. And then there are risks to others, mainly during the treatment interruption phase of the study, where people go off of meds and are no longer undetectable, and then basically U equals U no longer applies. So in this talk, I'm really going to focus on the first two of these, but happy to talk about the others in the Q&A. So the first set of risks is related to HIV cure treatments and age, and some safety concerns with that. So, these are all things that are pretty obvious, I think. Every medication, even prescribed medications from our doctors, that come from the pharmacy and are FDA approved, come with risk. But there's heightened risk with immunotherapy. And in particular, the regulators who really control the conduct of these studies are very concerned about liver toxicity. That comes up over and over, that damage to a person's liver is basically can be, can end a study. And, we only have one liver, and, it's bad if bad things happen to the liver. So that comes up as sort of really the most prominent concern, but there are all sorts of other side effects that drugs can have. Those are all when used in isolation, but as I mentioned, often we're combining different treatments for these trials. And so we don't really know what to expect when we're combining different treatments, and the side effects could in theory be worse or increased because of the interaction between different drugs. A lot of these drugs also take a physiologic toll on the immune system. So they can kind of ramp up a person's immune system, kind of cause a broader immune response and a stress response that, can be a strain on people's heart, on their lungs, on their brain, on their liver, on their kidneys. And then there are a number of additional considerations with what's coming in the future, which are cell and gene therapies. That are kind of unique to those things. So I'm not going to spend a lot of time on them, but those can kind of cause these, at least in cancer, which they were developed for, can cause these hyperinflammatory states that can really kind of exaggerate that stress response that I mentioned. There are also risks related to going off of meds. So these are some examples of what it looks like to go off of HIV medications in two different styles of studies. So let me start by talking about what's on the right side, these time to rebound treatment interruptions. This is when a person goes off their HIV meds, and we're simply looking at the time when the virus first pops back up. So typically these are a little bit shorter. They're, 2 weeks, 3 weeks, 4 weeks, 5 weeks, maybe 6 weeks. And as soon as the virus comes back, we say the virus is back, restart meds. And so that's like pretty conservative, generally pretty safe. I'll talk a little bit more about this at the end. And then on the left are what are called set point treatment interruptions, which is what we did in this most recent study that I described with AMFAR, where we allowed the virus to reemerge, and we weren't really focused on when it reemerged or how high it went. We were really focused on where it sort of settled into, and that's what's called a set point ATI. So we allow the virus to come back, go up often to very high levels, and then kind of see where it comes down to. Does it come down to 50,000? Does it come down to 10,000? Does it come down to 1,000? In order to do that, people need to be willing and allowed to be off meds for weeks or months. And so that's a sort of more intense treatment interruption. So there are lots of concerns that you can think of with these. Some are age specific and others are not. So there's a risk that when the virus comes roaring back, that people can develop kind of that seroconversion syndrome that some people have and just feel lousy related to that. There's actually no direct relationship between whether a person had that when they initially acquired HIV and whether they'll get it in a treatment interruption that I'm aware of. There's the physiologic stress of the virus coming back and the immune system responding to that. So, increased heart rate, maybe increased body temperature, increased respiratory rate, generally increased metabolism. There's concern about immunologic consequences, that a person's T cells might drop, for example. There's concern about damage to organs. So will that stress cause somebody to have like a heart attack or a stroke or hepatitis? This isn't common, but it's a theoretical risk. And the important thing with all of these risks is that we think that they all kind of just disappear after treatment is resumed. And so, as a researcher, I believe that if you do this in a controlled way, very carefully with good monitoring and great communication, that it can be done safely in most cases. But a question that's come up over the last few years is whether the consequences of these really long treatment interruptions. So this paper came out a couple of years ago, got a lot of attention. Many of you may have seen it. It was published in the journal AIDS. And this was a single center study looking retrospectively, so looking back at the last 20 years, and about 180 people who had participated in 10 trials involving treatment interruptions. They had 136 participants who actually stopped their HIV meds, and a smaller number, about 45, who didn't stop their HIV meds and were used as comparators. And they looked over a 20-year period in these people. And they looked for cancers not related to HIV, heart disease, infections, liver disease, kidney disease. And they found that the occurrence of this was higher in people who had participated in treatment interruptions. It was about 43% in people who had previously done a treatment interruption. This is years after the interruption, versus 23% in people who had not. On average, these events happened about 5 years after they resumed HIV meds. So it wasn't in the immediate term. And ultimately, their conclusion was that people who participated in a treatment interruption had basically fourfold the likelihood of having one of these events happen. What was this driven by? So it was really driven by a few things, and it wasn't necessarily the things that I would expect. So I would be worried about cardiovascular and neurologic things, but actually those were more common in the control group. the things that drove this risk in the ATI participants were cancer not related to HIV, liver disease, and kidney disease. It's hard to know mechanistically why that would be in a way that would sort of like spare the cardiovascular and neurologic things that as a clinician, I would actually be more worried about, that were more common in the people who didn't do the ATIs. So, my overall assessment of this is, there's a small study, retrospective, really some of the data was quite old. Like this was generations of HIV meds ago. So like in, early 2000s kind of generation of meds. And many people were off of meds for a really long time, like years. So, my assessment of this is, we all know that there are risks to participating in treatment interruptions. We need to be really thoughtful about this, and we need to sort of look at this with more modern data. But this wasn't sort of a, we don't have to worry at all kind of situation. So I think it gave me a little bit of pause. What about the brain? There's, a lot of concern, especially as people age, with cognitive function. We know that people with HIV are more likely to have mild cognitive issues as they age. This wasn't really looked at in a high amount of detail because there weren't a lot of people who experienced this in that other study. There is some good data from Thailand showing that short treatment interruptions don't really seem to have an effect on neurologic health, so stopping therapy for a few weeks. But there's not a lot of data on longer treatment interruptions, and I think that this really needs to be studied and should be studied. So ultimately, the result of all of this is that the most common solution to these concerns is what is shown on this slide, which is that the studies have a long list of inclusion criteria, and one of those criteria, often the first or second, is that the study should only enroll people who are younger than 65. And that's, kind of an easy solution. I'm not sure that's the right solution, but it's a very conservative solution and is the reality of what's happening. And so I'm going to take a couple minutes now and talk about efficacy concerns as well. So, some drugs work less well as people get old, and immune system modulating drugs are one of those classes of drugs, we think. We know that as we get older, our antibody responses decline, our T cell responses decline, even in people without HIV. And this phenomenon is called immunosenescence, which took me like 5 times to try to spell correctly. But it just means that the immune system gets older and gets maybe a little bit more sluggish as we age, and that's normal and fine. Historically, this has been best studied with regard to flu vaccine because we worry about influenza in people as they get older because it's more severe and it lands people in the hospital. And so there were some studies conducted in the 90s showing that flu vaccines simply work less well, especially in people who are older than 70. The result of that is that when people get older than 60 or 65, I can't actually remember now, we switch over to doing high dose flu vaccines, basically a double dose. That was based on some randomized clinical trials published maybe 15 or 20 years ago. And so there's concern that if we're looking for something to cure HIV that relies on an immune system response, it may be less effective in people who get older, and we may be less likely to see an effect. But not everything is influenza, and this study came out during COVID, one of the good things that came out of COVID, showing that actually, like, these are responses to COVID vaccines, the Pfizer COVID vaccine, and the response is pretty stable until age 80, where it just drops precipitously. And so, for flu, the cutoff would have been more around age 60, but for COVID vaccines, which I think kind of have the immunogenic potency that we're talking about with HIV cure stuff, the cutoff is very clearly 80, which is much higher than 60 or 65. So, I think maybe the mantra about looking at the flu responses isn't, necessarily the only thing we should be looking at. So I have a few summary slides for a couple minutes, and then we can have questions. But so the big picture, what are the important considerations? I think for safety, they're obvious. As people age, they often accumulate more medical diagnoses. That doesn't happen in anybody. There are, I have patients who are 85 years old who look better than some of my 5-year-old patients. So there are definitely concerns about toxicity of agents and drug-drug interactions that may be more important as people age, simply because people are more likely to have other medical problems and more likely to be on other meds, but that is not a universal truth. There are some concerns about efficacy. We want to make sure that we can optimize the good effects and avoid the bad effects. I think that before COVID, there was progress being made on this. Like for the AMFAR study, we had gotten the age limit up to 67. And the FDA was on board with that. But basically when COVID happened, everything sort of reverted to, everybody who's older is at higher risk of COVID. And we ended up not enrolling people above 65 for that reason. But progress, there was sort of momentum, I think, led by people on this call toward that. What do the guidelines say? So, the most important guideline for cure studies is this, which came out a few years ago, which laid out considerations related to treatment interruptions. And I looked at this carefully again this morning, but there's actually no specific age exclusion. It talks about people being otherwise healthy. And there's this really nice sentence. If a study only allows individuals with a very restricted age limit, may be difficult to enroll, might exclude a large proportion of the HIV-infected population, thereby precluding their contribution to and participation in HIV research, and would limit generalizability of findings to the broader population of people with HIV. So I think that's a beautiful sentence, and a sentiment that I really agree with, that I think needs to sort of be discussed in a more focused way. So my thoughts on this are that chronologic age is not the same as biologic age. There are some young 80-year-olds and there are some old 50-year-olds. I think that it's the responsibility of an investigator to assess whether age or comorbidities could affect the scientific integrity of a study. Like, do we think the therapy is not going to work in people over a certain age? I think it's incumbent on the investigator to have a real discussion about risks and safety with participants. It's also important that participants are willing to hear that discussion and are not, sometimes people want so badly to be in these studies that they don't even want to talk about the risks, which is also not good. So I think this really requires true informed consent. There's a lot in those 35-page consent forms that is not, in my opinion, useful, but this is something that is, in my opinion, important. And then I think, not at the individual level, but there are broader consequences that are relevant in terms of the context of the study, because these studies are so highly monitored and regulated that even a single adverse event can really basically end the study for everyone. And so a risk of being more liberal with the inclusion is that it may increase the likelihood that somebody will have an adverse event that could potentially end the study, even for younger people without comorbid. So that's just a background consideration. And then practically, my opinion matters very little, because ultimately the regulators are the ones who make the decision, and you often cannot negotiate. And so, I have been in meetings with the IRB, with the FDA, with funders, where I basically say, I want to allow people to age 67 or age 70 or age whatever, and they say no. And then I say, well, the CAB really wants this and everybody thinks it's important, and they say no. And so ultimately there is someone who has the final say, and those are the people who need to be influenced. And so, we really need more advocacy from investigators, but also from community about that. In the meantime, immunotherapy is really the core of cure efforts, and ATIs are not going to go away soon. because we don't really have good biomarkers. There are efforts to shorten ATIs to allow wider participation. I think this is really important. So I mentioned at the beginning that sometimes we do these very brief ATIs, and I want to spend one slide talking about that. These generally are more acceptable to regulators and may not actually require any age restrictions because they're so short and so highly monitored. And then I'm also involved with some efforts to model what happens during treatment interruptions to see if we can do a shorter ATI and say, based on 3 weeks off of therapy, whether this person would have controlled or not, been cured or not, so that we don't actually need to do the full 12-week or 16-week thing. There are also efforts to integrate non-treatment interruption options into some trials. So, for example, our CAR T-cell trial with UC Davis, initially everybody had to do a treatment interruption, but we actually modified the protocol so that there is a non-ATI group that is a little bit more liberal in terms of comorbidities, and you can imagine that working in terms of age. But ultimately, in order to test whether something really works, we need to be able to do ATIs. And so some of you may be familiar with this work from Matthias Lichterfeld and the Raygon group that we were also involved with, which is a really technical and interesting study looking at how the HIV reservoir changes over decades on HIV meds. And the important thing to look at is this middle circle here and this circle on the left, which look quite different in terms of the colors, right? There's a lot more red in this is good. In this circle on the left, which are people who've been on HIV meds for a longer amount of time. And this circle on the left actually looks a lot like the circle on the right, which are elite controllers, people who are able to control their virus without meds. What this suggests is that over decades of treatment, the reservoir may change and people may approach a cure, but it may take 30 years. So that means, for somebody to be on HIV meds for 30 years in 2023 means that they are older. And so this is a very important line of investigation right now. And in order to be able to understand this, we need to be able to take older people off of their meds. I think this can be done safely in a highly monitored way, but I just want to emphasize this is actually quite an important issue for the field. What does this look like in real life? These are some treatment interruptions that I've done here. So, on the left is a non-controller who stopped their HIV meds. They rebounded after exactly 3 weeks off of meds. And, we were able to detect when their viral load was at 200 and they came in the next day, their viral load was at 1,000 and we restarted them. So you can see this is quite a limited amount of time. They were basically off for 26 days. And then you can see someone who was a controller who was off of meds. It took 6 weeks for their virus to come back. It rebounded to a few thousand copies, and then they spontaneously came under control. And this person is sort of what we're looking for in cure studies, someone who's achieved maybe a cure state. So they were off of meds for a longer time, but it was highly monitored. There were no adverse consequences, and they actually controlled their virus. There have been studies that have looked at changes in CD4 counts and changes in inflammation, and we don't see them in these highly monitored ATIs, which I think is good news. We don't see major changes in our study. So in summary, immunotherapy and ATI studies are standard for HIV cure trials. Age limits right now are pretty restrictive due to a combination of both safety and efficacy concerns. From a safety perspective, there's nothing magic about the number 65. I think a comorbidity-focused approach is quite useful. From an efficacy perspective, we probably do need some cutoff, but maybe the cutoff should be 70, 75, 80. I think ultimately we need to know what's acceptable to the community. And if a change is acceptable and desired, we need advocacy among the investigators, but also with the regulators. And I actually think that there's a real need for sort of a commentary or position paper on this issue from the community that's focused just on this issue, to help move the needle if there is a desire to move the needle. So I will end there, and thanks for your attention, and happy to take questions. Thanks.
That was great, Michael. So interesting and really important. Let me just ask you a few quick questions before Michael goes through the chat. You know, a lot of these studies, well, all studies, as far as the FDA is concerned, and a regulator, they're done on a case-by-case basis. And there are some companies that are doing cure trials in older populations or sicker populations, which, you know, you're looking at the same sorts of issues. But, you know, so in other words, and I know the community needs to have a role in this, but we can't just say, as you said, 65 is a magic number. We need to do what you did with the gene therapy trial and say, okay, well, we can get away with this much here. You know, I mean, a lot of this stuff really moves in small steps as we gear up. And that I'm wondering if you can, we can keep your slides and share your slides with people, because they're extremely interesting about those adverse events. And it's really, you know, it's really important for people to realize. I mean, we had a couple of calls and people were like, we want to be included. We want to be included. So I thought it was really important for people to learn what these problems could be, you know, what the risk is here, especially into the future, you know, and you actually showed some really important data about that. So anyway, that's more of a statement than anything, but yeah, I don't know, I really enjoyed this.
I think that statement is very important because you know, we want to be responsive to the, you know, the desire of as broad a swath of people as possible to contribute to this. There are so many people who are so dedicated to the cause, but there's also sort of like a strategic decision to be made sort of on a study-by-study basis, related to both. So there's like the safety concern, which is one bucket, and then there's the strategic decision, which is kind of, is there a concern about efficacy and how do we get around that? In a larger study, you can simply stratify the analysis by age and say, did this work in people younger than X, and did it work in people who are older than X? But a lot of these studies for cures, you know, are quite small, you know, 10, 20, 30 people, and you can't really do that in smaller studies. And so there may be a strategic decision there. And then there's also the strategic decision of like, if we really think that people who are older or in whatever category are more likely to have an adverse outcome, like what impact could that have on the larger study's ability to kind of move the field? And so like, you know, as Linda certainly knows, like with the AMFAR study, in quite a young person, we had a serious adverse event. He was fine in the end, but it ended up almost sinking the study because we had to pause the study for so long that the people sort of behind that person in line didn't get the full intended immunotherapy treatment. It didn't matter in the end, but there are sort of those types of strategic decisions that need to be made.
So the last thing I'll say is I wanted to let you know and let the community know that we realize this. I mean, I'm always screaming about safety, but we're always having to worry about, we need this information for, you know, over 50% of people are over 55, and it's only going to get to be a larger portion of people over 55 as we go. So we need to do both things. We're actually the three MDC collaborating cabs, DARE, RID, and CRISPR. We actually have gotten contribution from their sites, their investigators, to do a social science study about what is important for people over 65 and that sort of thing. And there's absolutely no reason that we can't look at these you know, these balancing aspects when we do that paper. So hopefully, I mean, we were going to ask you to be involved in that, but so in other words, Corinne will be in touch with you and Steve about that.
Yeah.
Yeah, I mean, this is just so important, we think. All right, Michael, you want to shoot with the chat questions?
Sure. So the first question came back way at the beginning from Mark Gilbert, who was talking about 2 hospitalizations he had where he was off meds for 2 weeks each time, and yet he didn't see an increase in his HIV RNA, and he was wondering why that might be.
Yeah, I mean, everybody's different. And so, sometimes the virus comes back in 8 days, and sometimes it comes back in 28 days. And everybody sort of has their own tempo of that. So, that's not outside the realm of what might be expected. That's what I would say.
Okay, great. Shekina wrote, and Shekina, do you want to speak your own question, or would you like me to read it? I see you there.
Thank you, Michael. I'd be happy to share. So kind of in a real kind of quick, I know we don't have a lot of time. So in my particular situation, I was diagnosed in 85. I didn't start any kind of treatment until 96. And then through the course of that, through like say 2003, I was really on, off, and then on for about a year and a half, and then took about a 2.5 year break. So I've been off HIV medications more than I've been on during the 38 years I've been living with HIV/AIDS. So my question is, you spoke around the 5-year event, like after restarting, and typically you're showing that there's a 5-year event. Now, that's not held true for me. And I'm just curious if there's any data on other people who might have experienced the same.
Yeah, I think that's a great question, and sort of the limitation of that study that I presented is we don't really have a great sense of who these participants were and sort of what their trajectories were. And as you mentioned, especially in the early 2000s, it was common for people to cycle on and off of meds. And so it's really not known. I mean, I would really like to see that analysis that was done in that paper updated kind of with, a larger comparator group, including people who weren't participating in studies, but were maybe cycling off and on meds, and people who were diagnosed and treated starting in the more modern era, maybe 2010 and onwards, where it was standard practice never to go off meds. Because I think there are all sorts of different considerations. The meds back then also worked less well. So there are lots of different potential confounders that could have resulted in that observation. I don't think it's definitive.
Just to share with everyone, I've recovered very quickly from HIV encephalopathy. So that was my AIDS diagnosis in 2007. So pretty remarkable.
So maybe we better go to Matt and Derek, since we only have about 20 minutes left.
Okay.
Who wants to go first, Matt or Derek?
I just added Derek, and I need to find Matt on the.
He's on there. Well, Derek, why don't you go? I did see Matt. He's
Yep.
he's at
I'm here.
Yep.
I'm here. Can you hear me?
Yes. Why don't
Okay.
you go first, Derek? Take about 10 minutes, and then Maddie will go next.
Okay.
Sure. Thanks again, Linda and Michael and the rest of you for inviting me to be on here. I probably won't use up the entire 10 minutes, but I just wanted to give a perspective as someone who's been living a couple of decades with HIV and AIDS, and as someone who works in the field with those of us that are long-term. I have to say that there has been a very sort of interested and cautious optimism around ATIs, but and I want to underline the cautious. There are a lot of concerns about this, particularly when you're dealing with a community of folks that this is not their world. You guys are incredible advocates and activists that is supporting the effort and moving the science forward and developing better outcomes for folks. But most folks don't live in this world, right? And so I'm talking about people that sort of became HIV positive and still remember the messages of take your medication on time every time. And suddenly we're developing protocols that say, hey, we're going to take you off of all of your medication, all your HIV medications, and it'll be just fine. And mind you, I don't think folks are actually saying that, but that's the message that comes across. And we just want to see what happens. And that leaves folks feeling very vulnerable. What's happening? I think it's very similar to the message of U equals U sometimes, where we've been banged on the head around using condoms so much that the idea that me as an HIV positive person who's undetectable, suddenly I can not use a condom having sex is rather startling. So it's going to take a bit of time for the community to catch up with the messaging and the understandings of the science. And I think it behooves us to take the time with our various communities and talking to them about the development of the science. Like quite literally every single time you kind of have to go, and this is the history of how we got here. I seem to be well aware that there are definitely those folks and even older folks that are really rather, they want to be the renegade. And so every new thing that comes along, they would love to jump on it. But I see them, or I experience them more as outliers than the majority of folks who are just trying to keep up with understanding what the science is. They're quite appreciative of the work that's being done, particularly as we develop better medication and better strategies and of taking medications that lower side effects and help us to live longer. I am constantly having conversations with folks who talk about living on borrowed time, which is another both hope and challenge for folks. The idea that the medication I have was hard won and it finally works. And I'm, 50 and 60 and 70, and people are going, I didn't expect to get to 30. And now I'm at 65 and 70, and I'm contemplating retirement. Wow, let's leave it the hell alone. It's working. So and even that even comes across with some of the discussions I've had with folks around the injectables. The pills are helping me. The pills are doing the work that they're doing. I would also say that there continues to be concerns that are much more in the realm of conspiracy. So I hear a lot of folks that talk about this is, the cure is already there. This is the proof in the pudding. That they're trying these new strategies around the messaging that we've always been told. It goes against the messaging that we've always been told. They do have a cure, and they're just stringing this out for money and profits. And while I say that in a sort of mocking voice, we all know that there has been other instances where that is kind of a truism. You know what I mean? Look at our healthcare system. And so there are these hurdles that our community is coming to term with as they understand the new approaches and therapies that I think we constantly need to consider, and know that this is not a one-time message. This is an ongoing conversation. Much like we say when we talk about the informed consent process, it's an ongoing process. This is an ongoing process in educating our community about these newfangled ways and approaches. And it's really important to always add that we are not quite at the beginning stages, but as we talk about cure, we're talking about finding a cure and discovering a cure, as opposed to this is a cure or this is the product, which I'm constantly sort of navigating with folks who are saying, like, my doctor says that this thing is the next big hit, you know, and just like, let's mediate that message a little bit.
Okay. Now, before we go to Matt, I just wanted to say to everybody, we've been talking about advocates and activism and advocates. So I put a couple, I thank you, Alistair, for breaking up those different links for me. I put them in the chat because one is for, goes to the DAIDS CAB list of any sort of interest that you may have in HIV research, in vaccine prevention, treatment, cure research. There's information and contact information there. I gave you a specific contact name and email address for Allegra at the ACTG because they're desperate for advocates and activists to be involved in their program. So if you're interested, please get in touch with them. I also put a link in to TAG's cure page, which Richard, I think, is on the call, puts together, that is amazing for more information. And Michael, you're going to do the interactive how did you like the session at the end of this, right?
Says.
Okay, so I just want you to maybe stay for that so that you can tell us what you liked and if you liked it. And Maddie, take it away. I'm sorry to take from your time.
No, that's okay. I was already late anyway. But I guess I didn't miss much of, I did miss part of the beginning, but anyway, glad to be here. Thanks everybody for inviting me. I think a lot of you may know my story. I was going to tell the reason I'm interested in telling people more about cure research is because I was the second person in the Sangamo gene therapy trial, and I took a huge risk at that point. But I, spoke to a lot of people about the possibility of the danger of being in that trial, and at that point, I really needed something to boost my immune system, and we didn't think it would, but we hoped it would, the gene therapy. And it ended up, I was I got probably 20, 25 participants, excuse me, who had a sort of a side effect to the trial, which was, or I guess you'd call it a good outcome, which was our T cells doubled. Before I go on, even though I was in this trial as the second person in the trial, everybody in it was also on their antiretroviral therapy. So we were being treated at the time, and we had to be at that point. That we couldn't just give people gene therapy and assume that they wouldn't have any problems with their viral rebound or any sort of side effects. So our T cells, my T cells doubled, and what we've seen from that is that's been like 8 or 9 years ago. Somebody can correct me about when we did that trial. It's been a long time ago, And what's happened to me, and this is my story, it's happened to me is that I still have sort of a set point that's almost double of what I had when I entered the trial. So my T cells have never gone down any further after I was in that Sangamo trial. So something happened, and there's been some research to look at some, whether there is some kind of um, particular cell or something that caused that to happen, and we could discuss that another time or whatever. But when we first started talking about doing this series of webinars, my main interest was to just let people know, more that we were doing. That it was important that we needed to let people know that these trials were going on, because I think there was a small group of people that did know about them, but the wider community of long-term survivors and people who were aging may not know about them. So I wanted to make sure that we get in the beginning and sort of start educating all of us who are growing older that the trials are possible, and to make, we'll go into everything else over this series, but it's important that everyone is fully informed through the process of going through informed consent and all the way to the end of the trial. There's a lot we don't know yet, but I think there's still a possibility. And the most important reason that I wanted to talk to people about research in older people is that it happened to me, and it's happening to other people who are entering in peer research trials. Certainly not everybody, but I wanted to I think the main thing is to get the word out. So outreach is important. So that's all I'll say at this point. Linda, is Linda
Linda, you're
talking?
muted.
Hello, Linda.
Mhm.
I was on mute. I just said, Michael Llewellyn, I'm sorry. See if you can get to any more questions in the chat, and I thanked Matt Sharp.
Sure. There were lots of questions. So I'm trying to find one that might be easier to answer. The slate date, let me see. Derek, you had a lot of praise for everything you said. It touched a lot of people's hearts, what you were saying. So one of the questions was, well, what can we do to bring more people living with HIV into this group, to encourage more people living with HIV to become more comfortable and aware of the science underpinning these types of clinical trials?
So, I think that's one of the main reasons that we're doing these sessions. We people tend to be in their own bubbles, like Derek described. We're pure activists. We know who we deal with. That's who we deal with. But we've really made a concerted effort this time around to do pure community engagement when I say this time around, I mean in this year of funding. Last year, we did an 8-series session on gene therapy and tried to reach people all over the country. This time, we've not only done the same thing with long-term survivors in HIV research, but we really pulled in a lot of people that work with long-term survivors and not just cure research, so that we could cover the field a little bit more. And we hoped that people who were on the call would get, be advised that this was happening from the long-term survivor community, so that they could get plugged in with this, which is why I always try to put those links out about how to get involved with this. I guess that's all I have to say. And Michael Llewellyn, did you want to say anything about that?
No, I don't need to say anything. I was wondering if any of our panelists wanted to say anything to that. Derek or Matt or even Michael.
I had replied to the question in the chat, and it looks like a bit of a mess because I was writing really fast. But one of the things that I tried here with our local CAB here was just asking about what people thought, and develop some questions that our local coordinator could address for our team, or for my cohort. And as we and as we talked about those questions, I wanted him to, and he was doing it naturally, just sort of add in the connection with clinical research and research into the sort of work that they were doing with their doctor, and making that connection for them, like, this is how this works. And in clinical research, these are the sort of things that are important. And I let not I, but we just let folks who had developed more interest in finding out more to join in and find out more. I found that that's the best approach, because this is not everybody's cup of tea. And so just allow folks to develop their interest. And even for folks that this isn't their cup of tea, doesn't mean that they don't want to hear about the information. People are very curious about what's happening in this field, because it does affect their health and well-being, right? And so they will always want to know what's happening. And people respond to folks that are in their community because they know those folks and have long-standing trust with them. So you want to develop those sort of leadership in the community.
Can I add one? I'll just add one quick thing.
Sure.
I was reminded of Martin Delaney and the town halls we used to have live pre-COVID centuries ago, and how it would be amazing. I mean, we're trying to do these virtually now, and that's great. But I was wondering if there's some way that you could do a series of live town halls to get people interested again. I don't know, that it may be a wild suggestion, but it just seems like now people are moving back to what are face-to-face meetings, and the town hall with somebody that's really an effective speaker about the issue would get a lot of people interested. So that's my comment. Thanks,
You know,
everybody.
Matt, I do those in Baltimore, and we've actually done them in San Francisco as well. Sometimes in San Francisco, I'm virtual, but they're not. So I've actually learned so much from that. As you know, I mean, you and I have been doing those kind of meetings since the year of the flood, and there's nothing better.
Okay. Yeah.
Michael, Luella, you see anything else that is
Nope. People were agreeing with Matt and agreeing with Derek. Someone said, you can sign me up, Matt. I'm in. I think that's it. And since we're over the hour, we should probably cut the.
Yeah, 2 minutes. We're 2:02. If you could send us your slides, Michael, that would be great. We'll make sure Michael's great at making sure they get out, Michael Luella.
Yes.
And thanks everybody for hanging in there. We really appreciate you participating in this, because without you, it doesn't really make any sense, does it? All righty. Thanks so much. We hope to see you next time. The next call is on August the 15th. And it's about women living with HIV. So we hope that all of you will come and join us for that as well. Thanks again. Take good care.
Bye-bye.
Bye.
Bye. Thanks, Michael, and Matt, and Derek.
Thanks, everybody. Bye.
Bye, Linda. Bye,
Bye.
Michael.
Hey,
This is
Adam,
so
bye.
important for me. I love you all.
Bye.
Bye.
Thank you.
Yeah, I'm learning so much. Thank
Good.
you.
Great, Wanda.
Bye,
Keep it
Linda.
up. Make sure you're there next week.